Endomembrane trafficking allows for the transfer of information and eventual formation of new proteins in the body. Protein synthesis is a process in which a cell generates new proteins from the information stored in DNA found within the nucleus. The nucleus is the control center of the cell. It is surrounded by the nuclear envelope with nuclear pores that allow certain molecules to move into the cytoplasm. DNA, or deoxyribonucleic acid, holds the blueprints for genetic codes needed in the body within chromosomes. DNA is composed of nitrogenous bases adenine, thymine, cytosine, and guanine. These bases are connected to their matches through hydrogen bonds. Adenine pairs with thymine, as well as uracil within mRNA, and cytosine pairs with guanine. mRNA, or messenger ribonucleic acid, holds similar qualities to that of DNA, though it only has one strand compared to two, and replaces thymine with uracil. mRNA transfers information copied from a strand of DNA to continue the process of protein synthesis. Transcription is the formation of mRNA in the nucleoplasm. Transcription allows for a strand of DNA to be copied into mRNA by having an enzyme called RNA polymers separate the two strands of DNA. As the process continues, RNA nucleotides find complementary bases on this coding strand. Each sequence of DNA is measured into groups of 3, called codons. When the mRNA is assembled, it detaches from the DNA and exits the nucleus through the nuclear pore into the cytoplasm, where it joins with a large and small ribosome to begin the process of translation. Ribosomes are responsible for creating polypeptides in protein synthesis. As a ribosome reads the mRNA, tRNA, transfer ribonucleic acid, attaches to the ribosome. tRNA carries an amino acid, the predecessors to polypeptides. tRNA also carries a set of 3 nitrogenous bases that pair with the codon on the mRNA. These bases are called anticodons. As the ribosome continues to read the mRNA, it assembles the amino acids into the polypeptide chain. Post-translational modification is the process in which the now synthesized polypeptide chain is modified into a protein through the rough endoplasmic reticulum and Golgi apparatus. A lysosome clears the unnecessary code while the protein is carried out in a secretory vesicle through exocytosis. Free ribosomes can be found within the cytoplasm. Others can be attached to the endoplasmic reticulum. Proteins manufactured in the endoplasmic reticulum are modified and packaged for secretion. The endoplasmic reticulum chemically alters the proteins and transport them to the Golgi apparatus through transport vesicles. The Golgi apparatus is made up of several flattened membranous discs and chambers called cisternae. The vesicles carrying proteins enter through the cis or forming phase of the Golgi apparatus. They are then modified with enzymes. When finished, the product exits through the trans or maturing phase, which is most superficial to the plasma membrane. Secretory vesicles and lysosomes contribute to the trans phase. Secretory vesicles contain secretions that will leave the cell through exocytosis. Lysosomes are responsible for the basic cleanup of the damaged parts of the cell and secreting waste products during exocytosis. The vesicles travel through the cytoplasm and attaches to the membrane and ejects its contents into extracellular space. The plasma membrane acts as a semi-permeable barrier between the cell and the extracellular membrane. Due to this process, the membrane flow within cells allows for proteins to be formed and used in the body.